I'm Daniel Levine, and this is the Bio Report. Hibernation is not just a matter of deep sleep. Animals that hibernate are able to do so without suffering damage to tissue and muscle. Understanding the biology of hibernation can unlock potential insights into obesity, heart attack and stroke, muscle atrophy, neuroprotection, and longevity. Fauna Bio is studying genomic data from so-called extreme mammals and applying its proprietary AI platform to perform comparative genetic analysis to find gene disease links and leverage millions of years of evolutionary adaptations to identify new therapeutic opportunities for human health. We spoke to Ashley Zehnder, founder and CEO of Fauna Bio, about the types of insights that can be gleaned from the genetics of hibernating animals, the wide range of human health conditions that might be addressed with such an approach, and the company's deal with Eli Lilly to apply its AI platform to discover new obesity drugs. Ashley, thanks for joining us.
Yeah, thanks for having me. I'm excited to chat with you today.
We're going to talk about hibernation, Fauna Bio, and how it's applying AI to deep sets of biological data about hibernation to discover new drug targets for neurodegenerative, kidney, heart, and other diseases in humans. Perhaps you can start with the origins of Fauna Bio. Why did you create it, and what was the need and opportunity you saw?
Yeah, no, it's a great question and one that we get fairly frequently. Our approach is pretty different from a lot of approaches that other companies are taking, but it really comes actually quite naturally out of the backgrounds of its founders. So my background is in veterinary medicine. I'm a veterinarian by training. I did a clinical specialization in exotic animal medicine, which for those that may not be aware, that really incorporates anything that people keep as pets that are not dogs and cats. So I like to say I do lion or parrots and guinea pigs and snakes, but not lion, tigers, and bears, right? Then was fortunate enough to then go to Stanford and do a more human-focused cancer biology PhD. That really came out of an interest in from my clinical patients, seeing the broad differences in which species got different kinds of cancer, which species seemed to get very little cancer, and really wanting to understand more of the molecular mechanisms for that. So I was fortunate enough to get into the cancer biology PhD program at Stanford. And that really changed a lot of my perspective of recognizing that, most of human biomedical development is really focused on looking at humans and then mouse and rat models of human disease, which are really imperfect in a lot of ways. And there was really a pretty large knowledge gap in terms of the diversity of diseases and how they present in other species. And it was really then meeting my two co-founders at Stanford that really crystallized the opportunity that fauna is serving. So my CTO, Linda Goodman, has a background in human genetics, became really frustrated, frankly, with how hard it is to find which genes are driving human disease if you just consider humans alone. It turns out that's a much easier path if you look at genes that maybe haven't changed over hundreds of millions of years. Nature has selected those for a reason. Linda likes to say, if you mutate those, you're a dead mammal. And so, there's a reason why those genes are kept consistent. And so then we try to look at what those genes are doing in disease relevant states. And then my CSO, Katie Grayback, also has a human genetics background. But she also rotated in the lab of a hibernation geneticist named Sandy Martin at UC Denver. And that was a lab that was really looking at core mammalian genes through this lens of hibernation. And we'll get more into that, I suspect, in a few minutes. But it's a model of extreme mammalian adaptation that was really fascinating for the purposes of looking at how the body can repair itself from damage. So we realized, just collaborating academically, that the three of us had these three very important aspects to how you would start to translate these insights into novel therapeutics. My clinical translational background, Linda's background in human genetics and disease target identification, and Katie's deep expertise, not only in human genetics, but also in these extremely adapted mammals. And so we realized that if, there was a deep misunderstanding of what data were available in some of these species. So expecting people that are used to doing sort of human drug development to go out and look for these data sets and then know how to use them was kind of silly. So we realized if nobody else was going to do this, if we didn't. So we decided to give it a shot. But it was really kind of organically came out of our unique sets of expertise.
Hibernation's a lot more than just deep sleep. How well understood is the biology of hibernation?
It's really interesting. So both Linda and myself had backgrounds in, say non-human, either both clinical data and genomics data, and neither of us really appreciated the complexity that is hibernation biology. So we both had to learn a lot after starting this company. But hibernation is a trait that's been studied well back into the 1950s and 60s. It's a trait that exists across almost every major group of mammals. So there are even primates in Madagascar that hibernate pretty similarly to 13-line ground squirrels, which is the model we typically use. So it's a very broad trait that happens in mammals, which suggests that these animals are actually using highly conserved mammalian traits and not, say, traits that are specific to any one particular species. So the biology of hibernation is quite well understood, with the exception, I would say, of sort of what drives them into hibernation to start with. That's a little bit less understood, but people are certainly working on that. So the physiology is quite well described, particularly how protected these animals are from different states that mimic human disease. So particularly how they can repair damage to the heart, prevent damage to the brain, reverse key aspects of metabolism, which I think we'll talk about more in a moment. That has actually been well described physiologically, and to some extent kind of genetically. Where fauna has stepped in is in taking some more advanced omics techniques, looking at gene expression studies, RNA sequencing, some proteomics, and really applying those to understand more at a genetic level what's providing the protection that these animals experience during hibernation.
Well, what happens to an animal during hibernation? What are some of the physiological changes that take place?
Yeah, it's a great question. So people do like to think about hibernation like a deep sleep. We actually think about it more as a state of suspended animation. So during hibernation, the neurons in the brain almost fully retract. So if you look at the brain traces, they're flat. So these animals are clinically brain dead during hibernation. They actually go through these periods called interbout arousals, which is where they rewarm from being almost frozen. They go down to about 4 degrees Celsius or about 32 degrees, 36 degrees Fahrenheit, rapidly rewarm back up to normal body temperature in about an hour. And they do that every couple of weeks. And so that rewarming period is actually quite damaging to tissues. It's a little bit like what happens after a human heart attack or stroke. If you go to the ER and get your heart attack or stroke unblocked, you get this rush of warm oxygenated blood back into really almost every tissue in the body. It's called a reperfusion injury. And that can cause its own kind of damage. That's a kind of damage that we see in these animals every couple of weeks that they have to repair when they go back into their periods of torpor or hibernation. They also, their metabolism also changes quite dramatically. So they drop to about 1 to 3% of their normal metabolism when they're in this deep torpor. And that goes up to normal metabolism in that same hour time frame. My CSO likens it to going from a deep sleep to biking the Tour de France in about an hour. So really dramatic changes in metabolism. They also, so when they're up in that interbout arousals, when they actually will go into a period of REM sleep. So they actually will go through periods of what actually looks like sleep, just for that 18 hours that they're quote unquote aroused. I will remind people that even though they are aroused during hibernation, these animals are still, under a deep Wisconsin winter under the ground. So they don't eat for 6 months. They don't drink. They don't urinate, which has important implications for, say, kidney physiology during an entire 6-month period. So these are just some of the changes that happen during hibernation.
It's interesting to think about, because when I first thought about what you're doing, I assumed it would focus on maybe metabolic diseases, but as you go through this brain exercise, I mean, one of the things I learned that muscles don't deteriorate, they don't atrophy when an animal's hibernating. So, as you think about the wide range of possibilities, are there low-hanging fruit for you to look at? Are there some diseases that are more obvious targets for your type of an approach?
Yeah, there are some disease states that have been, very well described in hibernation. This resistance to ischemia or reperfusion injury is one that I mentioned before, but that's been studied in a wide variety of species and also in a number of different tissues. And so actually our lead program in terms of our internal pipeline comes out of our understanding of what happens in these animals during periods of low oxygen or hypoxia. And these periods of fixing the damage that happens after this reperfusion injury that happens every couple of weeks. And so what we're able to do with the platform that we have, which is called Convergence, because it merges human and animal data, is we're able to then map small molecule compounds to genes that are changing at these reparative time points. And that gives us a quick step up into building therapeutic programs that can mimic that natural approach. So
would that have implications in heart attack and stroke?
yes, heart attack and stroke, which are actually historically quite difficult development areas. So we're actually looking at these for kind of more chronic cardiopulmonary diseases. But yes, you can look at the same mechanism of action, but look at it in a broad set of diseases where you have either cardiac damage or ongoing cardiac injury. So it doesn't have to necessarily be applied to the acute damage models, although we certainly use some of those models for validation of those targets for sure. We also can look at changes in the brain, which I started to allude to before. So there are really dramatic changes that happen in terms of tau phosphorylation. So some of the early signs of Alzheimer's that we see, these animals phosphorylate their tau protein pretty similarly to what happens during early stages of Alzheimer's. And that is reversed every time they re-arouse, as well as reconnecting all those dendrites that I mentioned were disconnected when they're brain dead. So those all get put back together every couple of weeks. So there's some really dramatic changes in the heart and brain that have been really well described, not only in the 13-line ground squirrel, but in a number of different animals that hibernate that very clearly link to human diseases.
Humans don't hibernate. How translatable are the genes and the pathways that play in hibernation to human biology? Does this pose any particular challenge to what you're doing?
Yeah, no, it's a great question. So humans don't hibernate, but it was interesting. Linda went through this exercise when she was helping the Xenomia project at the Broad really characterize the ability to modulate metabolism across all of the 452 mammals that are in that Xenomia consortium. So this is a large genomic consortium that aligns the genomes of, like I mentioned, 452 mammals. So they were characterizing those mammals on the ability to use torpor or hibernation, and they set a metabolic cutoff rate of being able to modulate your metabolism by about 50%. And humans kind of missed the cutoff because the best example they could find of reducing human metabolic rate went down to 40%. So we sort of just missed the cutoff. But what's interesting is that, as I mentioned, there are primates that can hibernate very similarly to humans. And I would say that there's a couple of different aspects of what we do that give us confidence of the mechanisms that we see translate into humans. One is that, again, we're looking at very highly conserved core mammalian genes. So these are genes that are conserved not only in hibernators, but also, species like ourselves that are not hibernating. And these mammals are just using those genes in a slightly different way. So we're not looking at, say, specific hibernation genes or genes that just exist, say, in 13-line ground squirrels. These are genes that are broadly conserved across, most of the 452 species in that consortium. We enrich then for known human disease pathways. So we actually take data sets from humans with diseases. So say humans with, after a heart attack or during heart failure, or for the work that we're doing with Eli Lilly, looking at human metabolic disease. And we'll put that data in our platform so that we can start to directly compare how genes are changing in an animal like the 13-line ground squirrel, and do those same genes show an opposite effect in a human disease state. And so we can see those same genes functioning in a slightly different way. And then, of course, there's the validation, right? You can only do so much on the computer. And so we do then take both genes and compounds that we predict with convergence and test them in the most relevant human cell line models that we can find, and then go back and validate those in animal models of disease. So kind of multiple checkpoints, from just the basic conservation of those genes to how they're modified in humans, to also looking at humans that have mutations in those genes and seeing if we see linkages to the types of diseases that we're looking at. So we work really hard actually to make sure that we're looking at pathways that will function similarly in people as they do in the hibernating mammals.
You mentioned ground squirrels. Are there other animal models or ones that are better suited for what you're trying to do than others?
Yeah, it's a great question. We started working with the 13-line ground squirrel because it really was the de facto and best described hibernating animal model. There are other ones. So we are working with another group at UNLV that works with an animal called a tenrec. This is a very ancient type of mammal that also hibernates, but in a slightly different way than the ground squirrels. They're a little bit less well studied, and so we're having to build up some of the resources for that species. And we are also looking at some non-hibernating mammals. So one of our other collaborations is with a group at the University of Florida looking at spiny mice. And these are a Middle Eastern species of mouse that can regenerate many different tissues in its body, including the brain, spinal cord, skin, kidney, that are normally in us are not regenerative, but they can regenerate these tissues without any scarring or fibrosis. So we have what we call our dream team, our discovery, research, and emerging animal models team, which is really a cross-functional group of folks at Fauna that reach out to academic investigators and look at what are the emerging animal models, particularly of disease resistance. And then it really is a cross-section of, compelling animal biology plus the ability to actually get access to tissues to be able to design the studies we need. And so, we said we have an external innovations team that works with investigators to get access to those samples.
Your drug discovery approach is using AI. We haven't really talked about how your AI works, but one of the cases for AI-based drug discovery is not only the ability to find novel targets, but to do so cost effectively. Any sense of how your approach might compare to traditional approaches in terms of cost or time?
Yeah, no, I'm really glad you brought this up because this is actually something that we find pretty striking about the data that we work with. So, just as a point for comparison, our lead program, which is a small molecule that we found by looking at ground squirrel heart samples, we were able to discover that small molecule by just looking at 60 ground squirrel samples, where a comparative human study to try to find that target might have taken tens or thousands or tens of thousands of human samples, because there's not that, sometimes the differences between human populations are just not that big. Because we're working with animals that have very extreme changes in their physiology over a very short time scale, you can see those differences much more easily. So in kind of technological terms, there's just a much higher signal to noise. It's much cleaner data. We also are looking specifically at changes in gene expression at precisely the time when these tissues are repairing themselves. So as opposed to looking at some human genomics, if someone is kind of uniquely protected from heart damage, for example, or say ages but doesn't get Alzheimer's, you have to mine that whole genome and try to figure out what's different about those people. Here we're taking samples, say, directly from heart tissue and then looking at what genes are changing as the tissue is repairing itself. So it's a much easier bioinformatic question on how to do that. Where the AI really comes in is linking our fauna data to these very large external databases like the whole UK Biobank, other resources like this GeneNet. So 35 external large data sets that people kind of traditionally use for drug discovery, all that's put into a very large knowledge graph. And that allows us to then really quickly identify high priority targets, but with the lens of starting with our own data that's much more precise. And so we can oftentimes go from really starting data to, in vivo validation for targets, within, several $100,000 as opposed to maybe a couple million that you might need if you're taking a broader approach. We also kind of let nature do the work for us. So we don't have to set up billions or millions of cell line studies to generate the data to then do the AI on. We just have to get the right samples from the right species. And so the upstream data cost is much lower. And it's much more meaningful. So for all of those reasons, not only is the AI helping cost effectiveness, but also just the data itself is really helpful in that regard.
And at what point do you move from the computer to the wet lab? Is there some threshold you look for in hits, as it were, with a knowledge graph?
Yeah, so there's several different parameters that we use to prioritize targets. It's usually having a compelling, biological signal from the ground squirrel, whatever species we're starting with, seeing clear overlaps with human disease genetics or functional genetics in terms of looking at gene expression in human disease, having a clear kind of druggable hypothesis in terms of how we would move forward with those targets and are they druggable from a variety of modalities. The platform is modality agnostic, so we can be a little bit flexible there. And is there sort of any clear risk in terms of safety? So we have a lot of safety data put into the platform as well. And so once targets check all those boxes, then we start to design experiments to validate. If we can find good human cell line models, we like to start there because we can actually screen more targets through the in vitro models. But some disease areas, actually obesity is a good example, there aren't really good cell models of obesity. You really have to test in a whole animal. And so there we do have the capacity to do our own in vivo experiments. And we're building more capacity to do that every day. So a lot of the, even the early in vivo studies we'll do ourselves. But it really has to kind of check the boxes in terms of, say, clear biological signal, clear links to human disease genetics, clear safety signal, and really a clear druggable hypothesis.
I know that Fauna has announced a few partnerships. I do want to ask about those, but is Fauna planning on developing its own pipeline as well, or is the business built around forging partnerships?
Yeah, it's a very interesting question, and one that many platform companies have to define their business model, right, in terms of platform versus pipeline. I think we have always been interested in building a pipeline from the data that we have. We've always felt like if there are good targets and compounds that come out of this approach, we want to make sure that we are able to capture the value of those, as well as working with partners. It's been interesting building that pipeline. We do have a lead program and a secondary program that have come out of the pipeline. It's been interesting as we built capacity to push those pipeline projects forward, not only through validation, but also through lead optimization and, kind of early development candidate declaration. That capacity internally has then allowed us to then build more robust partnerships because we can take predicted targets much further in the pipeline because we built the capacity to do it ourselves. So in terms of partnering strategy, where we have focused historically on partnering are in indications that are, say, very large and complex and expensive to develop, or where there's kind of more risk in terms of maybe historical failures. So obesity certainly, fell in that pipeline. And we started doing partnerships in obesity actually as early as kind of early 2020, because this was an area where the 13-line ground squirrel clearly was a very unique model of metabolic flexibility. But obesity had just such a, at that point, very sordid history in terms of prior development, was very expensive to develop, needed very large clinical trials. It was something that Fauna as a company was not interested in building an internal pipeline in obesity. So that was a very natural place to start partnering. There are other disease areas. Heart failure is another one that can be difficult and has historical failures where we have some very clear data signals. CNS diseases, neurodegeneration are similar. So there are big disease areas where it would be nice to work with pharma early, where we try to build internal pipeline assets or where we have a mechanism or a target that clearly relates to a slightly more well-defined or potentially rare orphan disease population. Of course, recognizing that you still need a marketable indication, and oftentimes those rare orphan disease indications can give you confidence for a larger indication down the road. But we've tried to always be mindful of building a complementary approach between our pipeline and our partnerships, and we don't plan on doing a huge number of partnerships, but do the ones that allow us to build capacity in a new disease area or really show the breadth of the platform.
You mentioned obesity. I know the company announced a deal with Eli Lilly worth up to $494 million. Can you explain what you're doing with Lilly, but also what hibernation can teach us about obesity?
Yeah, no, two great questions. So with Lilly, we are really starting with fauna data and predicting really new targets that are not related to GLP or incretins for the purposes of obesity treatments. So very focused on obesity, really excludes looking at related comorbidities. So it doesn't relate to necessarily heart failure, renal failure, or disease, diabetes. It's really focused on obesity. We are looking at time points in the hibernation cycle where animals are very quickly increasing metabolism. So during those interbattle arousals I mentioned earlier, some of the current issues with the GLP-1 drugs are that, as many people know, you have to stay on those drugs or you will start regaining the weight. So, how do we fundamentally alter human metabolism to be able to make that weight loss more sustainable? Obviously, there are issues with patients losing some lean muscle mass on GLP-1 drugs, which is more of a problem for some elderly patients. So is there a way to rebuild skeletal muscle? This is actually an approach that BioAge is doing in its partnership with Lilly is kind of pairing a muscle, drug that builds muscle with some of Lilly's drugs. And then also really just looking at how do you control appetite, right? So there's a interesting time point before animals go into hibernation where there's plenty of food around. It's in kind of the mid-fall, but they stop eating about 2 weeks before they go into hibernation. And so what's controlling that hunger and satiety? So there's lots of different aspects of the hibernation cycle that are directly relevant to obesity, and it's really what we're digging into with Lilly.
I know the company is also working with NASA. What was NASA's interest in working with fauna?
Yeah, funnily enough, it's, in some cases actually kind of similar to what Lilly is interested in. So NASA is very interested in how to protect people that are spending more and more time in space. And some of the major issues with that are not only how do you potentially reduce human metabolism for longer term space flight, but also protecting humans from effects of things like low level cosmic radiation, effects of loss of muscle and bone, and kind of a disuse atrophy that happens in low gravity or microgravity. And so interestingly, there's some of the same pathways that we're going to be looking at for the on-Earth use cases in obesity, but then also in space, which is really interesting. So we have two active NASA-funded programs. One is called ASCRIPS, and that's really a program looking directly at are hibernators more resistant to the effects of low-level cosmic radiation. There have been some historical studies looking at much more higher levels of radiation, sort of like nuclear testing levels of radiation. This was, again, more in the 50s and 60s. They seem to be resistant to that, but we're trying to generate direct evidence that they have protection against the kinds of radiation that astronauts are more likely to encounter. And then there's a really interesting project that NASA has funded through their Innovative Advanced Concepts Program, or NIAC, that's funding the development of an animal unit, essentially, to go up on the space station. So we can actually take animals that are hibernating, put them in space for a period of time, and see how their physiology adapts to that and if they're protected from some of the effects that the astronauts are seeing. We realized early on that the actual problem with running those studies is that there was not an animal unit that can take those animals up in the space station, actually control temperature, and do the more complex physiological measurements that you would need to answer the question. So it actually boiled down to more of an engineering challenge. And so that work we're doing in conjunction with a group called BioServe in Colorado that does a lot of engineering contract work for NASA. And they're helping work with some of our team members to design that animal unit to go up on the space station. So that is work that is in phase one, hoping to transition to phase two funding early next year.
And how is the company funded today? How far will existing funding take you?
Yeah, so we have been funded through actually a combination of mechanisms. So we've been VC funded really from the beginning. We started out in Laura Deming's Age One Accelerator. That was our pre-seed round. And then been funded since then largely through a little bit more tech bio focused funds that are interested in really exploring novel biology and novel uses of, say, omics data. So our really anchor investors are True Ventures, which came into the company in early 2019. Also have invested in companies like Inveda Biosciences, which is looking at natural molecule discovery, and also Basecamp Research, which looks at novel proteomics and protein discovery. So we're sort of the genomics arm of that triad of companies. So VC backed from the beginning, but then also have gotten a little bit over $1 million in government grants for a couple of different programs, both our NASA programs, but also our therapeutic programs. And then obviously we had some partnering revenue last year from Lilly. So kind of a nice combination of funding to date. Right now, we didn't need to do a Series A last year because of the round that we did with Eli Lilly, but we'll end up doing a Series A at some point in the probably near future to fund our first therapeutic program and take that into the clinic.
Ashley Zehnder, founder and CEO of Fauna Bio. Ashley, thanks so much for your time today.
Yeah, good to talk to you, Danny. Thanks for the invite.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.